Hi, everyone. Welcome to our webinar. We'll get started in a moment. We'll give attendees a chance to come and join our room. Here, we appreciate you joining us today for our Earth Day themed webinar with Vernier and Bio-Rad. I'll be introducing your presenters here in a moment. Just to get some housekeeping things out of the way, we will be taking questions throughout the presentation, so feel free to either use the chat feature or the Q&A. We'll be monitoring both of those. Some questions will be answered in the background of the presentation, and then some we may choose to answer live, so we will definitely get through all of those. If we happen to not get to your question, we will definitely follow up after the webinar today. So it looks like we have a good amount of people in. We'll get started here. So today's agenda, we're going to do a quick poll that will ask you 3 different questions about your familiarity with Bio-Rad and Vernier and the grade level that you teach. Then I'm going to introduce your presenters. And then they will be doing some photosynthesis and cellular respiration activity with algae beads. So we'll be talking about that more in depth, and then we'll be doing a hands-on demonstration. We'll have time for Q&A, but we will again be answering those questions throughout the presentation, so feel free to use the chat and Q&A. And then I will be putting a link to where to find the recording of today's webinar after the session, but I'll put that in the chat closer to the end of the webinar today. And I want to introduce you to our first presenter, Sarah from Vernier. She has 15 years of teaching experience and was a professor of biology at the University of Incarnate Word. She earned her PhD in zoology from Oregon State University, and she's an advocate of science education and was involved in K-12 outreach activities throughout her career, including STEM summer programs for middle and high school students. And next up, we have Damon from Bio-Rad. He has a bachelor's in science degree, biology and chemistry from St. Mary's College in California. He taught high school in Portland, Oregon. He's a researcher at the Joint Genome Institute for 10 years, and he's been with Bio-Rad as a curriculum and training specialist for 10 years also. So Damon and Sarah are going to be your main presenters, and I also wanted to introduce you to John at Vernier. He's going to be helping to field your questions as well. So he has a BA in biology and psychology from Sonoma State. He also has a PhD in zoology from Oregon State University. He's a postdoctorate fellow from the University of Oregon, and he was an associate professor of biology for majors and non-major courses in biology and neuroscience and in active learning with emphasis on data collection and inquiry. So again, we are here to answer your questions, so feel free to put those in the chat or the Q&A, and I'm going to pass this off to Sarah to talk about spectral analysis.
All right. Well, we're going to be using a free app today called Spectral Analysis. And Angie is currently showing you our webpage. You can navigate to this page if you go to our main website and then just click on Downloads and select the button for Spectral Analysis. It's available for 5 different platforms. So you'll see under the name Vernier Spectral Analysis towards the right, you'll see the 5 different buttons, one for Windows, one for Mac OS. You'll notice the white button that says Chrome Web Store. That's the one that you want for Chromebooks. So if your students are on Chromebooks, you want to make sure that you use the version from the Chrome Web Store. But we also have it if you're on iPads. We have a version for iOS devices that you would get from the Apple Store. And then also, lastly, a version for Android devices, and that is the Google Play Store.
And I have dropped that into chat, so if you want to get to that webpage as well, it is located in chat.
Perfect. Thank you, John. All right, so we're going to be telling you today about a really cool lab that you can do using algae beads in the kit from Bio-Rad for photosynthesis and cellular respiration. It's an incredibly fun activity to do. This is one of my favorite kits just because the algae beads are incredibly fun, I think, for students. And we'll be using our GoDirect spectrophotometer. Sorry, I should say the full name, GoDirect SpectroVis Plus spectrophotometer. And we'll be taking you through some sample data. I'll actually take some live readings here and show you the prep as we go. But first, we're going to have Damon from Bio-Rad tell you a bit more about the kit and its history and the organism that we're going to be studying today.
So this lab is constructed on kind of two fundamental parts of the system. We have these freshwater algae that have been encapsulated into a biopolymer, basically alginate, that can then be dunked more or less into a pH indicator system that can tell you about kind of where carbon monoxide or carbon dioxide is in a solution. And using these two things, we can really easily monitor basically cellular respiration going on or photosynthesis. Bio-Rad designed this kit based upon looking at AP tests originally, like over 5 years ago, and realizing that half of the AP biology students, which should be your best and brightest biology students coming out of high school, were coming out not realizing that plants did cellular respiration. We know they got that in the theory portion of the class, but traditionally we've always run two separate labs, one for photosynthesis and one for cellular respiration. So we wanted to figure out how could we make a lab that could allow students to see this in one system. And so this is what we came up with. The beauty about this too, from a use point of view, is that these beads are really easy to use. It's definitely one of the easiest Bio-Rad labs to run. The algae that you're using is Cenedesmus obliquus, which is really nice because it occurs all across the Northern Hemisphere, so you don't have to worry about any kind of disposal issues. And then also the beauty about using these beads is you have them encapsulated in such a way that you can now put this in a spectrophotometer and don't have to worry about light scattering as it hits individual cells because they're all grouped into these perfectly sized beads that will basically sit in the bottom of your spectrophotometer and can allow your beam to shoot over the top of them. They have a shelf life of about 1 month after you activate them. And what that means is after you take them more or less out of your fridge, expose them to light for the first time, and then they have a 3-month shelf life from when you get them from Bio-Rad. And so when you get them, you'd want to store them in the refrigerator, and then you would pull them out and activate them by more or less washing them off, giving them some light for usually a day or two to allow them to kind of turn their machinery back on. So that's basically how these beads work. And then just to kind of give you a feel of where Cenedesmus is in the world, especially for anyone here to North America, if you use the Inaturalist.org program, you can actually look up where any organism exists. Basically, anybody with a phone or a microscope in this case can take pictures of an organism, upload it to a global database, and you can see where people see this organism. And so you could easily just punch in Cenedesmus, and you can see all the observations of where this thing exists in North America. So that way it also helps your students understand where these tools are coming from. This is something they could literally find in a freshwater pond near them. The pH indicator portion of this system is this really well-constructed indicator system that's very sensitive to pH change. And so at kind of atmospheric carbon dioxide levels in your room, it usually starts off in this kind of reddish hue. And then if, say, you have algae beads sitting in there that are in the dark and they're just dumping out CO2 because they're going through cellular respiration, it will quickly shift the pH scale down towards 7 or a little bit beneath 7.0, and you'll see that color shift to be a bright yellow color. Conversely, if those beads, you take them and you put them in the light, they're still doing cellular respiration, right? You've got to do that to live, but they're also scavenging that carbon dioxide up that's in the water and dumping O2 out into the water. And so it'll actually shift this pH up towards pH 9, where you get this really magenta purpley color. So it's a really easy visual recognition for them to tell what's going on. And if you've got a spectrophotometer, you can now track this really quantitatively, which is really, really cool, because then you can start talking about rate. And once you have that kind of nice tight quantitative measurements of rates on just a light and dark reaction, then we can kind of look at other things that might affect that rate. So besides just a light and dark reaction, within the kit, Bio-Rad also scaffolds you through a series of kind of investigations. And those you can see on this next slide. So After doing a light and dark reaction with your students, you could then have them, see what does light intensity do to those rates? Do those rates move at the same time? Does photosynthesis and cellular respiration, do rate shifts happen together, or do certain parameters cause them to move disjunctively? You could also test the type of light color just using filter sets. You could also use these systems and vary their temperature by more or less putting them in an ice bath or a warm water bath. The beauty about these cuvettes is they've got these nice little lids that go on them. So you can literally put them in a water bath and not have to worry about the system leaking. And then of course you could do the classic one where you build just a little mini ecosystem by, say, putting a freshwater snail or slug into your cuvette. And usually what the students will see then is that once you get a heavy breather in there, it's really hard to actually pH shift it upwards. You can also take these beads, and there is a de-beading solution that comes in the kit. And so you can actually kind of melt out a couple of beads and students can look at the algae underneath the microscope. So that way they can really see what they are working with. The kit runs about $145 for the AP biology version of the kit, which comes with the cuvettes that go right into Vernier spectrophotometer. You can also just buy a refill version of this kit for $119, and that's just the reagent pieces. So just the beading solution, just the beads, and the pH or the color indicator. That pH indicator, you end up getting about 500 milliliters of this. So since you're really not burning through a whole bunch of this in each lab station, you can actually take one kit and cycle it through multiple sessions of students, which is really nice because this is really a great kit as far as kind of bang for the buck goes. And what I always encourage people to do is figure out how to use the kit the first time using the full kit, and then in years going forward, get to the point where you're using the refill kit over and over. And this is a really easy lab to do this with because those cuvettes, as long as you don't allow them to kind of get beat up and scratched up, you can just use them the next year. And all you really need is just those refill reagents in order to run this again if you keep those plastics. For context with this lab, because this is one of the things that doesn't always get talked about when we talk about how to run a lab, because we're so focused on how to do the detail work, is with this one, I find it's really, really important and much more engaging for students to give them some sort of phenomenon of why they might care about the rates of photosynthesis and cellular respiration. And so this is one of the classic ones I use at national shows just because it's a really well documented case. But what I'm going to encourage you guys to do at the end is actually look for ones that are local to you. So I usually start off by showing the students this photograph and ask them, what's going on? Or actually first, what do you guys observe? And you pick up a couple things like, okay, we've got a bunch of dead fish. And then I usually kind of like tease out some more questions like, well, what do you think that water is? Do you think it's fresh? Do you think it's salt water? What type of habitat are we in? Just kind of trying to tease out any little things they can see, help them build those skills, those observational skills that some of them just need that teasing to get it out. And then once you get some of those observations down on the board, then ask them, okay, using those observations, what do you think might be going on? What might be causing a bunch of these dead fish to show up? And you'll get the whole variety of stuff, right? Everything from pollutants, when you're like, okay, and give me a mechanism that involved in those pollutants, or dynamite fishing, all kinds of things. But usually let them get a whole bunch of hypotheses out there of what might be going on, and then say, you know what, I want to add one more layer of information that I got about this situation for you guys to then whittle down some of those hypotheses a little further. So then I would show them the next image. And I would say, okay, that bag I showed you where all those dead fish were, that's actually this Atchafalaya Bay just west of where the Mississippi dumps. And if we look at that map, what do you guys see from the satellite image? And let them kind of come up with ideas like, oh, the water's kind of cloudy. And to some degree, you can see that the water actually has this kind of green tinge to it. And you can ask them, well, what do you think that might be? And then let them give out a couple hypotheses. And some of them will already kind of probably heard this story in the news at some point. So some of them will already kind of drift towards the fact that you might be having some sort of algal bloom due to nutrients flowing down the giant Midwest nitrogen pipe, also known as the Mississippi. And you could say, well, why is that a problem though? We've got all these nutrients flowing down. There's all this algal life blooming all over the place. There's tons of stuff to eat. What does algae do? It produces oxygen, right? So this seems like a good thing. Why would we get dead fish? And I'll let them, and that one's actually really tough for them because a lot of the times they hear this in the news, it doesn't go into the details of why this is happening. So usually what I'll do is I'll show them the next image and we'll talk about the dead zones a little bit. And so these dead zones occur in areas where you have these big kind of phytoplankton blooms, and there's a number of things that contribute to basically a dead zone. Some of it is that you've got all these things basically blowing up and living and then falling to the bottom where they're being broken down. And some of that breakdown process is also pulling down O2 with it. But then another major factor that can be involved here is that you have got this huge population now of algae that during the day is producing a ton of O2. But at night, they're only doing the cellular respiration part of their cycle, which means all of that O2 that was dumped out, they are now scavenging that up out of the water. And this is really hard on larger organisms. Small organisms can kind of, they can get by okay in lower O2 concentrations depending upon the organism, but you get to these larger vertebrates like fish, and they have a really hard time if you have an O2 crash at night that's partly facilitated by O2 being gobbled up by all of these O2 producers that are there during the day. And so we can say, well, this seems like a really important thing to understand then. How could we model this to understand these rates? Because these rates are obviously very important. What happens if the Gulf goes up in temperature? Does the rate of cellular respiration go up the same as the rate of photosynthesis goes up, or are they disparate? If they are different, this could be a really, really big problem, right? If cellular respiration goes up, say, twofold and O2 production only goes up 1.5, this is going to be really, really bad in the long run. And especially with global warming going on, we know these changes are going to be taking place. And so that's how I kind of set the field for them a lot of times to then jump into this exploratory hands-on lab. However, though, the Gulf Coast or the Gulf is a great, case study, but what's even more powerful to your students is something close to home to you. And I guarantee you, any place you're at in North America, unfortunately has a pretty good example of this close to you. So if you're in the Pacific Northwest, you've got the Hood Canal. If you're in California, you've got the Salton Sea. And pretty much any state, you will be able to find a pretty strong case study that can be close to home for the students. And so I would use that as a way to kind of set this up, to have them have a little bit of skin in the game of why they want to understand how these systems work. I have a couple of kind of tips and tricks for this lab that I've learned over the years from using it that I'm going to share with you guys that aren't in the Bio-Rad manual. Bio-Rad manuals, remember, we write them so that they are bulletproof, but being a former classroom teacher, sometimes I'm willing to give up a little bit of bulletproofness to kind of make things happen faster. And so I like to share those with folks that are using these kits sometimes. So let me give you a couple of these really, really quickly. So the first one is the preparation for the pH indicator. You're getting a 10x concentration of this pH indicator, and you're supposed to basically dilute it out with water and then let it sit out for a couple days on your desktop to allow it to basically adjust to your atmospheric carbon dioxide levels. I am super impatient, as I imagine most instructors are. And to me, I was like, when I first saw this, I was like, oh my God, 3 days, I can't do that. I can barely do 2 days. What happens is if I'm just waiting for CO2 to access this, this is just a surface area problem. So what I'll do is I'll dilute my stuff up to 1x, get 2 cups and just pour it back and forth until I see it stop changing color, right? Because I'm just facilitating that surface area reaction. Once I have my pH indicator, I will take my beads that I've got in my dark vial, I will wash them with water, with distilled water, as the Bio-Rad manual suggests. And then what I like to do is I like to put all of them just into a small Petri dish and then cover them with the 1x indicator to then put them in a light source. And the reason I like to have them in the 1x indicator is I can tell when the beads have started to do photosynthesis because the indicator will turn purple. And that allows me a quick visual reference to say, yep, my beads are happy, ready to go. If you don't have LED lights and you're using like one of those old school kind of like heat lamp generating lights, you definitely want to make sure that you've got a heat sink between your beads and that light source, because this algae, even though it grows in ponds that are relatively warm, it is not right at the surface. It's down a little bit where it's not getting fried by the heat. And if you put heat on it, you can accidentally kill them. So really important to use a heat sink if you're going to use a hot light with these. The other trick I like to say is I take the little transfer pipette that comes with the kit, and I cut it off so that I'm getting rid of the small beveled piece of the barrel. And this means now I've got basically a transfer pipette with a barrel that's just barely bigger than the beads. So I can actually easily suction up those beads out of my Petri dish and then add those to the cuvettes. And so if we go to the next slide, you'll kind of see how I arrange this for my students. I could build individual stations and put them out for them, but I'm also lazy on top of not being impatient. And so what I will do is I'll put up together a self-service station for the students. I'll take my transfer pipette, I'll pull up 10 beads and I'll put it in cuvettes for them. So that way they don't have to sit at their desk and move the beads around. When I have them do that, I just lose time. And so what I'll do is I'll make a light dark reaction for them, put it in the box. I will then basically fill a cuvette up with the pH indicator. I'll fill a cuvette up with water, and then I'll have one that's just labeled waste. And what students do is they come by this box at my desk and they basically just pick up one row, and those are all the reagents they need to run this lab. Next to the box, I'll have some transfer pipettes and some aluminum foil for them, and then they're ready to go. And then when they're done with the lab, they'll come back and then just re-rack this, which means that for the next class, all I need to do is have some more cuvettes with pH indicator in there and some more with water in there, which makes this really easy to kind of turn over quickly. The beauty about this too is if I'm running this lab, say on a Monday, and I have another class I want to use this on Wednesday, I just leave these in the box, leave this box in place with the lid off, where it gets a little bit of ambient light, and those beads will be super happy come Wednesday to then run the lab all over again. Let's go and go to the next one. We've got these color cards that come with the kit that students, if they're not using a spectrophotometer, can basically do a visual inspection of how quickly their cuvettes are changing. Just so that I don't have to buy these every year, I'll just put them in Ziploc bags, and that way they're basically protected against liquid spills, and it's a lot cheaper usually than my laminator at most schools. The other trick I like to use, especially for the spectrophotometer, is on this next slide, there is a series of cuvettes that I'll make so they can see basically the endpoints of the pH indicator spectrum. So that far left one, I'll take that pH indicator and I'll just put a little drop of vinegar in there, which will shift it all the way down. And on that far right one, what I'll do is I'll just do a little spray from the whiteboard cleaner or any sort of basic cleaner I have around, and it'll shift it really far up. And so that way now I've got basically the far ends of the spectrum. And then I'll also make a cuvette just of the pH indicator as it was at atmospheric CO2. And this is nice because if I want to give them a quick standard curve or have them build one, we've got 3 data points now, far into the spectrum and right dead in the center of the spectrum. One of the other, or 2 actually things to be aware of with this, depending upon your spectrophotometer, you might want to use 1 less bead than Bio-Rad recommends. They recommend using 10 beads, and this is just, because we're a base 10 organism. But the bummer is with this, you get a 3x3 square on the bottom of beads and then 1 sitting on the top each time. And so sometimes what I'll do is I'll just add 9 beads to it. And instead of using 10 beads and 1 milliliter of pH indicator, what I'll use is 9 beads and 900 microliters of the pH indicator. The other thing to be aware of is if they have the cuvette sitting up straight like this, or even on their side, what they'll start to see is as those are in the light, you'll get a halo effect around the beads. You want to make sure that they shake the beads first, shake that pH indicator first, before taking their readings. Otherwise, their readings will be pre-skewed because you don't have an even diffusion of what's going on in the solutions. And that's basically the biggest kind of tips and tricks I've learned from using this over the last couple years. And so I will, at this point, turn it over to Sarah to show you guys this in action.
All right. Well, I put on my PPE because we do recommend that students wear goggles when they're doing this prep. I have my algae beads that have been under a light. And I also have pre-prepped while we were waiting. I pre-prepped, or while Damon was speaking, I pre-prepped my cuvettes. Now I'm going to walk you through the Vernier adapted instructions for using our spectrophotometer. You can certainly create your own hybrid protocol for between what the tips and tricks that Damon told you and the instructions that we provide for using our spec. You certainly can adapt things and make it work for your classroom. But I've already prepped a blank for the spec. I've already prepped the test cuvette because when we were actually creating this protocol to adapt, we found that phenomenon that Damon mentioned where the color change is happening right around the beads, we found that if we use a pipette to siphon off or to draw off the indicator into a test cuvette from the cuvette that has the algae beads in it, that it actually mixes it as mixes it really well actually as it's being drawn off and then put into the second test cuvette. So we'll walk through those instructions as we go. I'm going to take us now to spectral analysis. All right, you should be seeing my screen with spectral analysis. And when you first launch the software, you're going to see a screen that asks you what type of experiment you're going to do. For this particular experiment, you are going to use the absorbance versus concentration mode. This would also be the same mode that you would use if you were doing a Beer's Law experiment. And I know you might be thinking right now, wait, if I'm doing concentration, that doesn't sound quite right because this is a timed experiment. But I'm going to show you how you can change this so that you can, instead of concentration, you can use duration for your timed events. All right, now I'm going to connect my spectrometer. And I'm going to connect it by Bluetooth. So I've just turned on my spectrophotometer. And you can see now it's showing up here in the software. I'm going to connect to it. And the SpectroVis Plus can be used with or without a USB cable. So if you prefer to just plug it in on USB, you can absolutely do that. If you prefer to use it with the battery and actually have it on Bluetooth only, especially if you're using something like iPads, that's incredibly convenient to use it on Bluetooth. Now it does have a warm up for the lamp. So we're going to let our lamp warm up first, which I know a lot of times students get impatient at this part, but this is also a great time for them to prep a different part of the experiment. And I'm not following our instructions exactly because I've pre-prepped, just because we have a short time together today. I've pre-prepped some of the steps, such as creating the barrel pipettes and marking them, and creating those cuvettes that I mentioned earlier with the codes on the lids. So the way that we've adapted this, you only need to use 3 cuvettes per group of students, 1 for the algae beads, 1 for your blank, and then 1 as your test cuvette that you'll be moving in and out of the spectrometer. When I created our instructions, it did not occur to me to only use 9 algae beads as Damon suggested. So the reason that we have you move the liquid is so that an algae bead doesn't accidentally get above the beam in the spectrometer. All right, so now we can put in our blank cuvette for calibration. So I'm going to calibrate. You always want to make sure that your cuvettes are wiped clean and that your clear, the Bio-Rad cuvettes have a side with a texture and then 2 clear sides. So they have 2, in one orientation, they have ridges that make them easy to hold onto, and then the other orientation, they're smooth. You want to make sure that the smooth side is oriented in the beam path, the light path of your spec. All right. So we're going to select a wavelength next using our predetermined wavelength for this lab, which is going to be 550 nanometers. I'm also putting in the cuvette that has our test solution in it, our indicator. All right, and now you're ready to collect. So you can see that spectral analysis gives you a little breakout window that has the full spectrum, just for reference. You can either keep that on or turn that off. And now we're ready to actually collect some data. So let's get our first data point, which is going to be our test solution. All right, so we're going to start. Actually, before we do that, I'm going to quickly rename our data set. So I had mentioned that we can rename, we can change the concentration mode and tell it that we're going to use duration instead. So our first, the run that I'm going to do first is under the light So I'm going to call our data set light. And then I'm going to go here to concentration, to the column. And I'm going to click those 3 dots for column options. Select column options. And now I'm going to change the name of that column to duration and the units to minutes. And then simply apply. All right, and now we can collect our first data point. So we're first going to hit collect. We're going to give it 10 seconds to equilibrate, even though the sample has been in the cuvette holder for a few seconds while we were renaming columns. We'll give it a few seconds, and then it's pretty stable. We'll go ahead and keep that data point, and this is at 0 minutes. All right, so we've kept our first data point, and now I'm going to take you over to, so I'm going to stop sharing and take you to my other camera view. All right, so you're looking at the SpectroVis Plus. We've got our test cuvette in here, but what we need to do now is we need to transfer that liquid back onto our algae beads. So this is our algae bead cuvette. We're going to use our pipette for the indicator. Move this out of the way. I just gently draw it off. And then place it onto the algae beads. Try not to get too many bubbles. All right, and now that cuvette with algae beads is ready to be placed under the light. So I'm going to put it under the light source. My personal tip on this lab is to use a sheet of aluminum foil underneath the, so I just keep a sheet of aluminum foil handy, and I set it underneath the grow light, and then I lay the cuvettes on top of it. That seems to help make sure that all surfaces of the algae beads are getting some reflected light on them. And now I'm going to set a timer for 5 minutes. And we'll let that sit and start photosynthesis. All right. So do we have any questions yet from our audience?
I was just going to point out one thing that you did instinctively that might need to be vocalized, which is with the cuvettes, you saw that Sarah oriented hers like flat and then laid it down onto her aluminum foil. You definitely want to do that because then it kind of spreads the beads out more and they're able to get even more light down and penetrate to them. Because sometimes if you do it up vertically and you've got the light coming down from the top, the light just doesn't penetrate as well as when the cuvette's on its side.
Thank you, Damon. You know, I realized I was doing that and I wasn't even thinking about it.
And the other thing to think about there too is if you've got your light reaction going and you've got your dark reaction, you want to have them side by side underneath your light source, just to minimize, right, the variable of temperature that might take place from light coming down. So it's always good to actually have the students try to generate that kind of concept of like where they should be putting that. And a lot of times if you just ask them, where should we put the light and dark sample? Should they be away from each other, close to each other? You know, they'll make the right decision, but you kind of want to trick them into making that decision on their own to some degree, or lead them down that path.
Yeah, I actually left, in fact, I can show you while we're waiting for our 5 minutes. I left a couple of samples last night under the grow light, and I did exactly that as you described. I had one that was wrapped in foil, and right next to it, I had the light sample. And here's our light sample after sitting overnight under the light, nice and purple. And then I'm going to unwrap this one. One caveat with our software, it's difficult for students to do both runs at once. So it's hard to switch back and forth. So the way that we've written it up is we have them do one run and then do the other run. But what I would recommend if you were doing a class with multiple students running this, is you could always split the class and have one half do the light run and one half do the dark run, and then switch if you have time. With a typical college lab, you'll probably have time to do that. With a high school lab period, it depends on your scheduling. But here's our dark run is incredibly yellow, and our light run is very, very purple. That's about as purple as I've seen it get.
Yeah, that one's very purple. But it's great to have a set like this when the students come in the classroom, just so they can see the far end of the spectrum shift. If you're not going to build those doped up ones with a little bit of, acid or base to it. And I actually like what Sarah does with this, because then they actually see it within the system and go, okay, the beads are there. So they kind of have a mental expectation of where theirs are headed.
The light that I prefer to use for really any photosynthesis experiment, but definitely for this one, would be, as Damon mentioned earlier, an LED grow light. They're becoming incredibly inexpensive. They are very easy to obtain on websites. You can find them usually at regular stores, like household stores. And you can also order them easily online, but they are very energy efficient. They don't put out a lot of heat, and they provide exactly the right wavelengths that you need for photosynthesis. And they really make a difference when you're trying to do an experiment with photosynthesis, to have that incredibly bright light that your cells need for those processes.
And in the right spectrum, which is big, because a lot of times with like high schools, sometimes I've had them just use their cell phones, which do work. You can get them to work, right? But it's not, these tend to be not quite in the right spectrum. So they'll go a little bit slower. But if you're in a pinch, this is something you could use, but you definitely want to use Sarah's trick then of having a foil bottom that then the light is shining on to get more intensity on the beads.
Yeah, what we have found in some of our investigations, we did a few investigations comparing different light sources for photosynthesis, and the source that we found to really be the weakest was compact fluorescent bulbs. So these are mostly the swirly ones that everybody switched to for quite a while, because they were very energy efficient. But what we found when looking at the full spectrum of those bulbs was that they are usually in the blue, they're either very weak or non-existent. And they tend to be actually mostly towards the middle of the spectrum, kind of in the yellow green, and they don't tend to have a lot of wavelengths for photosynthesis. So that's usually the worst performers were always under the compact fluorescence. All right, so my 5 minutes is up. My watch just buzzed. So I'm going to grab that sample back. I'm going to switch back to our software view here. All right, I'm just transferring, as I showed you earlier using the pipettes, I'm just transferring that liquid back into that test cuvette. So bear with me while I do that.
Hold on. I've got one question here that came in, and that's, when you mentioned the blank cuvette, does that mean empty? So what are you using for the blank?
Oh, sorry. That was a big oversight on my part. The blank cuvette has distilled water in it. So you'll want your blank cuvette to just have plain, clear water. All right, I've placed our test cuvette in. We're going to give it 10 seconds to equilibrate. Seems like there might be a bubble in there. Hang on. I'm getting kind of a funny reading, so let me double check, make sure I don't have anything on my surfaces. All right, there we go. Okay. All right, looks pretty stable. So I'm going to go ahead and keep that. That's our 5-minute point. And now we can transfer that liquid back for another 5-minute incubation. All right, I'm placing the algae beads back under our light source. And I am using my, my aluminum foil trick. All right, I'm trying to think of any other tips that we might suggest for this lab. Your students will get faster, I think, with each run. Sometimes the first couple times transferring the liquid out, and of course, certainly if you use Damon's tip of just doing the 9x9, or the 3x3 algae beads in the bottom. The big thing is to just make sure that they resettle when you tip the cuvette upright. You want to make sure that they're laying completely flat on the bottom, and that may take, you can always take a cuvette and gently tap it with your finger, or you can tap it on a surface if you need to move the algae beads around gently without getting any air bubbles in there. All right, do we have any other audience questions?
I'd say one other tip just to bring up, just because I've seen a handful of people do this, is that they'll prep the stuff and then they'll keep the beads in the fridge. You want to keep these at room temp before you go to work with them, because otherwise everything's running slow when students start with it. And so it'll take quite a few minutes before you start to see changes. And so when you prep this, just leave it out at room temp. These are perfectly fine at room temp for a month. And so that's where they're happiest. Yes, John.
We had a couple questions. One was, can you use an older spectrometer to do this lab if it doesn't have Bluetooth? And then the other one was, could I use a colorimeter?
Yeah, that's a great question. So if you have one of our older specs, they should work fine for the protocol. If you have SpectroVis Plus, or if you have the original SpectroVis, those steps should work quite well. So you could do this prep. John, remind me, the beam path of the original SpectroVis was a little higher, correct? So you might want to have a little bit more fluid?
Yes, that is entirely correct. One of the great things about the Go-Direct SpectroVis Plus is that the beam height, so where the light goes through the cuvette, is much lower, whereas the SpectroVis Plus, the beam height is actually much higher. So you would need to make sure that you actually have the cuvette filled up with at least, we used to say about 2/3 full, but you should be able to just check it by filling it full of a colored buffer solution and just verifying that you see a change. The only other thing would be the older SpectroVis Plus, and the SpectroVis can only be used over USB. So just know that you can use spectral analysis via USB as well.
And if you have to use a larger volume, you just want to, if you're trying to keep the same kind of rate structure, you just want to adjust the beads accordingly. And so the ratio is 1 bead to 100 microliters. And so say you had to go up to 2 milliliters, you're now going to want to use 20 beads, or just expect that you're going to run, at about half the rate as the normal ratio.
Thank you, Damon. I was actually just about to ask you that. If you have to adjust the volume, what do you do on the other side? And
Yep,
thank you, John, for remembering the question about Bluetooth, because I had forgotten that half of the question. That's great. We're getting close to our second run.
And if you're teaching much like an advanced class, a lot of times it is great to have students figure out where the beam path is and come up with a procedure for doing that. And most of them can figure that out if you kind of, give them a couple crumbs to kind of get there. And the easiest way is to, like John said, is have a colored liquid solution, like you could use the pH indicator, and add, 100 microliters at a time until all of a sudden you're seeing a change in the absorbance. And then usually most students will be like, okay, that's where it's at. And then you might want to ask them, well, how do we engineer the system so that we know we're never going to accidentally hit the meniscus, And so maybe add 5 to 25% above, where that level was that hit. And so bringing them through that process can be really helpful, because I guarantee you when they go to a real lab, they might have to actually do that with some of their experiments to figure out where that beam path is on an older instrument.
Yeah, that's a really good suggestion. All right, it looks like it's time for another run. So let me go back to our spectral analysis screen. All right. I'm going to do our quick fluid transfer here. Damon, while I'm transferring the fluid, you had mentioned that you definitely don't want to put these under a hot light. What about the sun? What about, we have a lot of teachers ask about using a sunny window for an experiment like this. Is that safe to do with these beads?
It is, depending upon how hot that sunbeam gets. And so I would say if you're in, Arizona or Southern California, you might be a little wary about putting them in the blazing heat of your window. I've only seen a problem once where I was trying to do them outside in Southern California. That I actually had them kind of kill the beads. What you could do to hedge against that, if you want to use a windowsill, is you could leave a couple ice packs on that windowsill, like if you have flat ice packs, and then put them on top of that. It'll kind of keep that temperature a little bit more mild, or just cool down that bench top over the windowsill before you put them on there.
All right. So we've got our 10-minute point.
And actually, since we've got that picture of kind of the full spectrum there, one thing that I love doing with spectral analysis and Vernier's different types of spectrophotometers is usually having students figure out where we should be taking the reading for different labs. But this is one of the only ones where I don't recommend that, just because this pH indicator system is relatively complex. And so there's kind of multiple peaks that they might choose. And the reason that we've picked this specific wavelength is because you can see a shift in it that correlates really tightly across this really wide color change. And so it's not going to be intuitive for them, because they'll see a higher peak and be like, oh, let's pick that and follow it. And then they'll all of a sudden won't be able to correlate it really tightly. And so this is one of the few labs I wouldn't have them look at the spectrum and then pick where we should be doing the absorbance, just FYI. Or if you really like to watch them struggle, you can let them do that and have a couple groups, pick different places in the spectra and then see which one they can actually follow the best. But that's for a much more advanced class, probably.
Damon, there was a question about, I realized that one of the other parts of the question that John just asked us, I completely forgot, and that was colorimeter, using the Vernier colorimeter. John, remind me, our colorimeter has it has an LED that emits, I want to say it's pretty close to 550. Is it 565?
Give me 1 minute to look it up right here. There's the Go-Direct colorimeter and the normal colorimeter. They both use 4 wavelengths, and I'll be able to get you the LEDs that they use here in a minute. 430, 470, 565, and 635.
Do you think that one of those would work, Damon? Do you think the 565 would work?
My gut feeling is the 565 will probably trace it pretty quickly or pretty closely. And so you most likely could get away with reading this on that colorimeter.
I don't know where the beam path is in the colorimeter. I think it might be up a lot higher. So once again, same thing. You would have to make sure that you had enough solution in there to get it to work.
Right.
Yeah, again, you can feel free to, if you need to adapt the instructions that we are providing. And it may take a little trial and error. It would make a great student project, I think, if you have, as Damon suggested, like it's always, I think, fun for some of the more advanced students to actually have to figure out some of the instrumentation specifications and figure out how best to optimize the lab.
I mean, as a former researcher, I can tell you, like, my first 2 years at the national labs, there was a large portion of my role that was figuring out how can we actually use the instruments we have to do the readings we needed to do. And so having them build that kind of troubleshooting skill set is really valuable.
Yeah, and if you find that you're struggling with the equipment that you have, you can always give Vernier a call. My department is tech support, and we are always happy to chat with instructors and figure out if we can help you adapt a lab protocol. So never feel like you can't call us, because we actually love talking with educators. So please don't hesitate. Or you can email us at biology@vernier.com. I know we're getting towards the end of our scheduled time. It's almost time to take another data point. We should be able to squeeze in another data point before we finish today. Possibly another 2, we'll see.
And one of the things on troubleshooting too is remember there's always multiple answers or protocols that will work to get you what you're trying to do. So I remember the first time I think we started playing around with this lab on Vernier's older SpectroViz, and that beam path was much higher. Me and John kind of gone back and forth about different ways we could do this. I think one of the first things I did was actually build just a little cardboard platform I could stick in there and put on top, right? So that way I didn't have to change my volumes. But then changing the volumes ends up being a lot easier than having to try and dig that cardboard piece back out. So there's always multiple ways to kind of find solutions. And so if you've got the time in your class to have students come up with those, it's really cool to allow them to see that science isn't just about one answer, especially when it comes to the engineering portions of science, is that there's multiple ways to do it. And you usually end up picking things for a multitude of reasons. A, that it works, and then B, that it's easy on you too.
All right, we're about to do our next sample. I'm going to switch my camera view. I keep forgetting to switch my camera view so that you can see the sample go into the spec. I always like to keep something like a Kim wipe nearby to wipe the sides of my cuvette as well, just in case my handling of it gets messy. Sometimes I will accidentally smudge the side, so it's always I find it a good lab practice to always have a wipe nearby that I can do a dry wipe on the
I have to admit, that's one of the things I love about the GoDirect SpectroViz Plus is that because that beam length is so much lower down, that even I've had students kind of manipulate the top a little bit too much, it really minimizes the chance of a smudge being down where the beam is coming through. But like most spectrophotometry labs with your students, one of the things you want to remind them, from the get-go is kind of where to handle the cuvettes.
All right, this is our 15-minute mark, and our data are sadly not going in the direction that I had predicted. And I have to say, I just ran this right before our webinar, and I'm going to give it another, I wiped it just to make sure. I'm trying to figure out if I might have gotten lint or something on it. No, it's about the same. All right, yeah, our data are not making a pretty linear function here. We are providing some sample data from the last time we did this workshop. I stayed late and I did a full run of both the light and the dark in one data set. So if you desire some sample data to use with your classes, you can certainly have access to that spectral analysis file. It is a spectral analysis file, so it needs to be opened in the software. So you'd want to launch spectral analysis and then use the file open pathway to open that.
This lab comes with those color cards. Another way that's good to kind of scaffold spectrophotometry into a unit is to have them actually take a picture of their cuvettes against the color card, build basically a rate study using that, and then compare that in contrast to what they get from the spectrophotometer, right? Because it helps them kind of build that mental model of that shifting color is really what that spectrophotometer is picking up. And so if this is one of your first spectrophotometry labs, I might do that kind of double data set with them. And the cool thing too that I've noticed about having them photograph their cuvettes against the color card is if you have them take a reading based upon looking at it, take a picture of it, they'll get slightly different readings based upon the way people see different color wavelengths. In fact, I actually see it differently between my left eye and my right eye because my left eye doesn't pick up as much yellow. And so my data is actually skewed if I use my visual versus my camera versus a spectrophotometer, which is really interesting because it helps them understand where different types of errors come from in processes when it comes to quantification.
Any final comments, Damon?
I think we covered most of the content, so I think we did a good job in that regards. I'd say the biggest thing to remember once again is with this kit, you can really stretch it over multiple classes, which is one of the reasons I really, really like this, because those beads stay alive for a month. So you can literally just kind of shift it through a bunch of different classes, which makes this a really good bang for the buck.
One thing I didn't mention earlier, but you're depending on your light source, in the classroom, you can adjust the times of this lab. So if you're finding that the color change isn't very much, if it's not happening rapidly enough, you can always extend the incubation period. So you can do 10 minutes. If you need to, you could do 15 minutes. If your students are all trying to share one light and you have to have the light backed out so that it's not as intense, that's a good way to adapt it. So you can play around, just like we mentioned that you can alter the volumes and the number of algae beads. You can also very freely alter the amount of time that it sits under the light. I wouldn't go in too large of an increment at once. You don't want it to accidentally go zooming through the whole run if you have an incredibly bright light, but you can certainly adjust it longer or short. You could even try shorter. I think when we were creating these instructions, we were trying to maximize the time in the laboratory by using a pretty intense LED light. But you could certainly adjust as needed to match your light source.
And if it's your first time running this, what I would do is get your light sources that you're going to work with, set up, 2 or 3 sets for yourself, and then do them under your light source and just adjust the distance from the light source for 3 sets. And that way you can kind of figure out, what's my rate under each one of those, and then pick the one that's going to work best for your classroom time period. Melville.
We have a question. What is the shortest overall duration for students to see the change? We only have 44-minute class periods, so is it tight to get done in just one class period?
It's really going to depend on how strong your light source is. And so for a really intense light source, it will shift all the way out to purple within 15 minutes usually. If I'm using a cell phone to do it, it's closer to 25 minutes to see the full shift out towards purple. And that's where the cell phone is almost laying right on top of the cuvette. So it's usually easy to hammer this out in a 45-minute classroom easily.
All right. Well, thank you everybody for joining us today. Thank you to our partners at Bio-Rad for being here for this webinar to celebrate Earth Day with us. And thank you to all of our participants.